اي جي ولازم تكون انتي بودي سبيسيفيك للانتيجين مش كده؟ تكونها عليها معقده اخرى اسمها كلاس سويتشنج شوت او دي مش بتاعتها دلوقتي دي العمليه اللي بشرحها لكم خلاص كده.
Yes, we can hear you.
Okay, let's start then. Okay, let me just present. Okay, I hope guys you can see my screen.
Yes, we can.
Okay, so let's start with the case number 3, which is about the liver and the gallbladder. So let's start. We have the learning goals, which involve the anatomy of the liver, the histology, the blood supply, the key functions of the liver, and how and where is bile produced, and the hemoglobin metabolism, excretion, and what is jaundice. Some, just briefly know the difference of the types of jaundice, the causes of jaundice, and all of that. Then we have the gallbladder, the anatomy, the histology, the function, the composition of bile, the function of bile, and how is the secretion of bile regulated, and the understanding the formation of bile salts from cholesterol, and all of that. The enterohepatic circulation specifically. Okay, so let's start first with the anatomy of the liver. So as we all know, the liver is the largest gland in the body, right? It approximately weighs 1.2 to 1.5 kilograms in adults. It's wedge-shaped and it has 4 lobes, the right, the left, and the quadrate and the caudate lobe. Out of them, the right is the largest one. And typically it also has some other divisions, the quinoid segments, and it's usually divided into 8 segments, which we will discuss in the upcoming slides. Then regarding the covering of the liver is covered by an outer serous layer, which is derived from the peritoneum, and it is covering the entire liver except for the bare area of the liver. I will show you the photo in the next slide. And then we have the Glisson's capsule, which is the fibrous inner layer. So the Glisson's capsule, it's covering the entire liver, including the bare area of the liver. So the outer layer is not covering the bare area, but the inner layer, the fibrous layer, is covering the bare area of the liver. So in this photograph, you can see the left lobe of the liver, the right lobe of the liver, and the bare area of the liver. And this is the anterior view of the liver. This is the posterior view of the liver, and from the posterior view you can see some structures just in between the junction of the right and the left lobe. This is basically in this porta hepatis, or this opening, you will find the portal triad. What is the portal triad? The portal triad is the I would say the entrance into the liver, or just an opening into the liver from which different structures are protruding, which include the portal triad. What is the portal triad? The portal triad is the common hepatic duct, the hepatic portal vein, as we all know the famous name by the portal vein name, simply, and then we have the hepatic artery proper. And at the posterior side, you can also see the cystic artery, right? So the main structures passing through the opening is the portal triad. So now in this picture, you can also appreciate the ligaments, the falciform ligament. Then you can see the quadrate lobe, which is the lobe. The inferior one is the quadrate lobe. The superior one is the caudate lobe. The caudate lobe is also referred to as segment number 1 in the quinoid classification, and it's posteriorly present. So we will discuss those segments. Then after that, you can see the inferior vena cava. You can see the fibrous appendix of the liver. Even the falciform ligament, it contains the round ligament, right? So these are the important ligaments. We also have the hepatoduodenal ligament, which is connecting the liver with the duodenum. We will discuss the ligaments in the upcoming slides. So that's it about this picture. Let's move on to the next slide. So as I said, we have the Coinat classification. It divides the liver into two functional lobes, right? The left and the right lobe. Now, these lobes are further divided into eight functional segments, and each segment, this is very important, that each segment is supplied by a biliary triad, a vascular biliary triad, which includes the hepatic artery, the portal vein, and the bile duct. So this is very important. And how you will classify or how you will call them by the right and the left lobe is by the segments being involved. So what segments will be in the right lobe and what will be in the left lobe. So regarding the right lobe, you have from segments 5 to 8. Segments 5 to 8 will be in the right lobe. In the left lobe, it will be from segments number 2 to 4. So that's it. And segment one, as I said, the caudate lobe is separate. It usually itself is a lobe, but you can even you can just attribute it to the left lobe. But once you are talking generally as the right lobe, the left lobe, the caudate lobe, then the caudate lobe itself is a separate lobe. But if you are talking about the Coinot classification, then it will be part of the left lobe. So over here, I hope you can see the segments of the liver. This is how the liver is divided. So as I said, the first is the caudate. So you forget that it's at the posterior side. Then we have segment number 2, number 3, and then we have 4, which is divided into two parts, 4A and 4B. This is how just they classify them. Then we have 5, 6, 7, and 8. Sorry. Yeah. So 4A, 4B, 5, 6, 7, and 8. So these are the parts. So now, as I said, which one from 5 to 8 is the right lobe? So from 5 to 8 is the right lobe. So this will be the right lobe, this division here. This is the functional lobe. And the left lobe will be called from, 2, 3, 4A and 4B. This will be the left lobe. But usually, anatomically, once you see the liver, you see that this is the falciform ligament, and you just divide it upon the basis of the falciform ligament as the right lobe and the left lobe. So this is just the main difference that you need to know, and you need to make sure of that so that you don't get confused in the exam. They might ask you about the Coinot classification, but the general anatomy of the liver, as I said in the first picture, the falciform ligament, the right lobe, the left lobe, the quadrate, and the caudate lobe. This should be very clear to you guys. You guys should not make a mistake in that. Regarding the Coinot classification, just have a general overview that these are the segments and in which lobe you will classify which segment. And each segment is supplied by a hepatic artery, the portal vein, and the bile duct. So that's it about the Coinot classification. Let's move on to the ligaments. As I said, the falciform ligament. Before talking about the ligaments, let's talk about the location of the liver. So the liver is present towards the right side. It's under the diaphragm in the right upper abdomen, and it's extending from the fifth intercostal space to the right costal margin in the midclavicular line. So I hope we all know what's the midclavicular line. So once you start from the sternal notch, you start counting the intercostal space. So it extends from the fifth intercostal space to the right costal margin within the midclavicular line. Then the ligaments, we have the falciform ligament. The falciform ligament is the ligament which is connecting this ligament, this one over here, the one in between this one. So this ligament, what is the function? It's connecting the liver to the abdominal wall, the anterior abdominal wall, and it is dividing the liver into the right and the left lobes. As I said before, this ligament is very important. So from anatomical point of view, so you know that it's dividing the liver into two parts, into two halves. Not two halves, two parts, you would say, because the right lobe is the larger one, and it contains the round ligament of the liver. So this is the importance of the falciform ligament, and very important is that it's connecting the liver to the anterior abdominal wall. So that's how the liver is attached to the abdominal wall by the help of the falciform ligament. Then we have the hepatoduodenal ligament. It is basically connecting the liver to the duodenum, and it contains the portal triad, the hepatic artery, the portal vein, and the common bile duct. Clinical correlate at your level, it might not be that important, but you can just know that why is the hepatoduodenal ligament important clinically. Basically, whenever there is liver hemorrhage or there is excessive bleeding within the liver, how do you stop the bleeding? You temporarily clamp the hepatoduodenal ligament. So once you clamp the hepatoduodenal ligament, you are stopping the hepatic artery from bleeding, or if the bleeding is from the portal vein, so you are stopping the bleeding from these two vessels. So this is known as the Pringle maneuver. So whenever there is excessive hemorrhage or bleeding within the liver, what do they usually do is that there is a maneuver called the Pringle maneuver. What do? They clamp the hepatoduodenal ligament so that there is a prevention of bleeding for a specific period of time, and then they surgically correct it. So that's about the hepatoduodenal ligament. Then we have the gastrohepatic ligament. It is basically connecting the liver to the lesser curvature of the stomach. So, and it contains the gastric arteries. So this was about regarding the ligaments. Now about the vasculature of the liver. Now the liver has a dual blood supply. We all know that, and it's by two different vessels. Regarding the arteries, you can say that we have the hepatic artery proper, okay, which is a branch of the common hepatic artery. So this is a picture. You see, this is the abdominal aorta. So from the abdominal aorta, you have the celiac trunk or the celiac artery. You can say that. Then after the celiac artery, you have the common hepatic artery. This is the common hepatic artery where my mouse cursor is. Then you follow this common hepatic artery, and it's giving off a branch which is called the gastroduodenal artery. This is just a branch being given off from the common hepatic artery. But the order you need to remember is that the abdominal aorta, the celiac artery, the common hepatic artery, which is then becoming the hepatic artery proper. So this hepatic artery proper is the main artery, which is supplying blood to the liver. Okay, so this is branching off from the celiac trunk of the abdominal aorta, and it is supplying only 20 to 40% of the blood of of the liver. So out of 100%, the 20 to 40% is being supplied by the hepatic artery proper. Regarding the portal vein, it is supplying 60 to 80% of the blood. Now, where is it coming? Where is it forming? It is forming at the junction of the superior mesenteric vein and the splenic vein. So let me show you the picture. Yeah. So over here, you can see this is the superior mesenteric vein. It is going upwards, and this is the splenic vein, the thick one. As you see, this is the splenic vein. So both of these are joining, and these are forming the hepatic portal vein. So hepatic portal vein is then going and supplying to the liver, right? And over here you can see the cystic vein supplying the gallbladder. So it is formed by the union of the superior mesenteric vein and the splenic vein, and it is collecting blood from the gastrointestinal tract, spleen, and the pancreas. So you see all of these vessels, all the blood that is being drained off from them. So the arterial blood is coming to them, and then it's going back right into the venous supply. So all of this blood is coming from here, and then there is the splenic vein, which is bringing blood from the spleen. So you have blood from the spleen coming, you have blood from the intestines and the pancreas coming through the superior mesenteric vein, and all of this blood is going into the liver. Now you see that this is the venous blood, right? Now, one of the functions of the liver is detoxification and removal of chemicals or waste products from your blood. So this is how it does it. It's taking the residual blood of your GI tract, your spleen, your pancreas, and it's getting this residual blood and it's going into the liver. And then it's being detoxified within the liver. So this is one of the very primary functions of the liver. So once this blood is entering into the liver, then you can track it back into the liver, and we will discuss specifically the histology that what's happening there. Then regarding the veins, we have the hepatic veins, which are draining into the inferior vena cava. Above you have the inferior vena cava, so the blood which is coming into the liver, it will go back towards the heart by the venous supply, so through the inferior vena cava. Then regarding the lymphatics which are supplying are the celiac lymph nodes. You can just remember them by the name. Okay, so this was regarding the blood supply. Now let's move towards the histology. Now we all know the liver, and the liver has some hepatic globules, and we all have heard about them. What's so special about them? Basically, these are sheets of connective tissue, and they are dividing the liver into small hexagonal units, as I said, and all of these, they have a central hepatic vein in the middle. And at the vertices or the vertex, you have the portal triad. What is the portal triad? Portal triad contains the branch of the hepatic artery proper, the branch of the portal vein, and the bile duct tube. Okay, it's a ductule because it's a branch of the bile duct. Then each portal triad is accompanied by some other lymphatic vessels and branch of the vagus nerve. So this is very easy to remember. You see this. Now this structure is a hepatic lobule. Now you can appreciate the six sides. That's why it's a hexagonal structure. And in the center, you have the central vein where the blood is draining. So the flow of the blood is towards the central vein, right? It's towards the central vein. And the flow of the bile duct is towards, the bile is towards the bile ducts or the bile ductules. And it's opposite to the blood flow, but it's parallel to the blood flow. This is very important. Okay, so they are going side by side, but they are in opposite directions. So you can see at the periphery or at the vertex, we have the hepatic artery, the portal vein, and the bile duct tube. Now over here, you can also see these are different hepatic globules clustered together, and all of these they form the liver, right? And within these hepatic globules, I hope you can see these these skin colored or peach colored cells. What are these cells? These cells are the parenchyma of the liver, or you can say the hepatocytes, right? So these are the hepatocytes within the hepatic globules, and the hepatocytes are the parenchyma of the liver. You guys might have studied the parenchyma of the lung. So same as that or similar to that, we have the parenchyma of the liver, right? Now this structure over here, if you see, this is a triangular structure. Now if you have some adjacent hepatic globules together with each other, you draw a triangle through the central veins of all of the central veins. So you will have a lobule which you can call the portal lobule. Why are you calling it the portal lobule? Because it's connecting by the central veins, and at the center you have the portal triad. That's why it's called the portal lobule. And once you connect the two central veins together and you draw a rhombus type structure, it will be called the liver acinus. But the most important for you guys is the hepatic globule. And you guys need to know what is happening within the hepatic globule. What is the histology? What are the components of the hepatic globule? What is present at the periphery? Where is the blood going? Where is the bile being formed? Right? So we will discuss that right now. Okay. As I said, the parenchyma is mostly consists of the hepatocytes, and it contains large amounts of smooth endoplasmic reticulum, which is involved in detoxification. I told you the function of the liver is detoxification, right? So the hepatocytes, they contain smooth endoplasmic reticulum, which helps in detoxification. Now you have two surfaces of the hepatocytes. You have the basolateral surface and you have the apical surface. Now you need to understand which side is which, okay? So that you can correlate with the function which is being carried out on each side. Now the basolateral surface of the hepatocyte is the side which is facing the sinusoids. Sinusoids are the fenestrated, you can say the capillaries, right? And the apical surface of the hepatocytes is facing the lumen of the bile canaliculi. So the apical surface is towards the bile canaliculi. The basolateral surface is towards the facing the sinusoids, right? So you can remember it by ABC, A, B, C, D. So A, B is the apical surface towards the bile canaliculi, A and B. Then the hepatocytes, we know what is the function of the hepatocytes. They are producing bile, right? They are making bile. So hepatocytes, they are making bile and they're secreting the bile into the canaliculi, and these secretions are flowing parallel to the sinusoids, but in the opposite direction of the blood, as I told you. And from the bile canaliculi, they will go into the intrahepatic bile ducts. From the intrahepatic bile ducts, they will go into the common bile duct, and they will eventually deliver the bile into the duodenum. The hepatic sinusoids are the large capillaries which are lined with the highly fenestrated endothelial cells. As I said, there are a lot of fenestrations present. Why are there a lot of fenestrations there? So that there can be proper filtering of blood, so that the waste products, the foreign materials, bacteria, red blood cells, anything can be filtered easily. Then blood flows through the sinusoids and empties into the central vein of each lobule. So let's see the picture. It will be very easy after that. So you see these are the hepatocytes, right? Now this is the sinusoid. Yeah, so this is the sinusoid and these are the hepatocytes. So the basolateral surface is facing the sinusoids. right? This is the basolateral surface, and the apical surface is facing the bile canaliculi. So these are the bile canaliculi in between. These are the bile canaliculi. Now this is the sinusoid. The blood is going towards the central vein, or you can say in this direction. Now you can see different structures, sorry, different cells or different components present within this image. So we will talk about them one by one. So we have the Kupffer cells, which are specialized macrophages. We have the ito cells, or the stellate cells. And then we have the perisinusoidal space. Now there is a space which is in between the hepatocytes and the fenestrated endothelium. This space is called the perisinusoidal space, and it contains the ito cells. These ito cells, they store vitamin A, and once they are activated, they make extracellular matrix. And once they start making extracellular matrix, they make fibrin. So the function is that they are important in fibrosis of the liver. Whenever there's damage to the liver, inflammation, fibrosis. Inflammation leads to fibrosis. So what is causing the fibrosis? The ito cells. Where are the ito cells? Ito cells are present within the perisinusoidal space. You will get definitely an MCQ about the function of the ito cells or the stellate cells. Then you have the Kupffer cells. The Kupffer cells are present within the fenestrated endothelium, or within the lumen, right? So you can see these green structures. These are the Kupffer cells. Now, the Kupffer cells are, as I said, they are a type of macrophages which are present in the sinusoid, and they phagocytosize foreign particles, bacteria, damaged old blood cells, right? So all of these, foreign particles, bacteria, they are being phagocytosized by the Kupffer cells. And as I said, the plasma filled space is the space of DC, or the perisinusoidal space. You can call it the two names, and they have the stellate cells or the ito cells, which will store vitamin A, and they are the main source of extracellular matrix production in liver injury, in the formation of scar tissue during fibrosis. So now we will talk about the zones of the liver. Within the hepatic lobule, you have zones of the liver. So you have zone 1, zone 2, and zone 3. Zone 1 is called the periportal zone, and it is the best oxygenated zone because it is nearest to the portal triad. I will show you the picture. Over here, you can see. So this is zone 1, this is zone 2, and the third one near to the central vein is zone 3. So zone 1, zone 2, and zone 3. Zone 1 is the highly oxygenated zone because it is very close to the portal triad and it's receiving direct blood supply from them. So it is the most resistant to ischemia, right? What is ischemia? Ischemia is reduced blood supply to any tissue of the body. It can be any tissue. It can be cardiac tissue or any other muscle. So whenever there is ischemia, this zone is the last one to be affected, right? So it's highly enriched by blood. So we know that it's the best oxygenated and it is the most resistant to ischemia. But the very important is that it has the highest amount of blood supply, right? So any chemical or any bacteria, any virus present within the blood will firstly affect the first zone. So, right, this is how you can correlate or make sense with the things. Okay, it is the best one to be oxygenated, or it's the best one with the best blood supply. But the drawback is that anything which is coming with the blood will affect it firstly. So, for example, you have viral hepatitis. Viral hepatitis is within the blood, the virus. So it will the periportal zone will be the first one to be affected. And any other toxic substance ingestion which is ingested into your blood, which you are ingesting and it's in your bloodstream. So, for example, cocaine, right? Any drugs, any other recreational drugs you're abusing, all of these drugs, they are being absorbed in your blood, and they are affecting the periportal zone as its first priority. Then we have the second zone, which is the intermediate zone. We all know whenever there is a counting 1, 2, 3, second one is always the intermediate zone or the middle one. So second you can remember by the intermediate zone, and it is affected in yellow fever. This is enough for you guys to know that it's affected in yellow fever. You do not need to go into the details. What is yellow fever, or what are the clinical features and all of that. Then you have the last zone, which is the pericentral zone, or the pericentral centrilobular zone, which is near the pericentral vein or the central vein, you can say. Peri means around, so it's around the central vein. So that's why you can even call it the centrilobular zone. Now this zone is the least oxygenated one, right? And it is the most susceptible to ischemia. So it's most susceptible to ischemia. It's the least oxygenated zone, and it's the most sensitive to metabolic toxins like ethanol, halothane, rifampin as a drug, acetaminophen. So all of the toxins which are metabolically produced, or the toxins which are a byproduct or waste product of the metabolism, are affecting the pericentral vein, the zone around the pericentral vein, which is the centrilobular zone. So this is the main function, and it has the highest amount of cytochrome P450. It's the enzyme of the liver, which is important in in metabolism of different drugs. So the highest amount of cytochrome P450 is found in zone number 3. So zone 1, the best oxygenated, affected first in viral hepatitis or ingestion of toxic substances. Zone 2, intermediate zone, affected in yellow fever. Zone 3, centrilobular zone, least oxygenated, most susceptible to ischemia, and most sensitive to metabolic toxins, and has the highest amount of cytochrome P450. So this is a picture, zone 1, zone 2, and zone 3. These are the 3 zones. And this is the central vein. So blood is flowing in this direction. Then, regarding the functions of the liver, you just need to know, you do not need to go into the details of the functions of the liver, because each function of the liver has a good amount of physiology involved in it, and you can read about it if you have free time. But just to give you guys a general overview, that what are the main functions of the liver. So liver is a very famous gland in our body, and it produces albumin. So albumin production is one of the core functions, right? What is albumin? We all know it's protein which is keeping fluids in the bloodstream, which we will discuss in the future. That what is the function of albumin in bilirubin metabolism? Then it is usually preventing molecules to leak from from the it's keeping the fluids in the bloodstream from leaking into the surrounding tissue. So this is the main function of albumin. It also carries hormones, vitamins, and enzymes throughout the body. So it's a protein that is present within your serum or within your blood, which is binding to different hormones, vitamins, and molecules, and it's helping them in transport. It's basically a transport protein. So who is making this transport protein? Liver. If there is anything wrong with the liver, albumin will be decreased. Then, we all know, very common function of the liver, bile production. What is bile? It is a fluid which is very important in digestion and absorption of fats, which we will discuss in the upcoming slides. Then, as I said, detoxification or filtering of blood. All the blood that is leaving the stomach, the intestines, will pass through the liver eventually, right? So all of this blood is filled with toxins. For example, if it's filled with toxins or waste products or byproducts or harmful substances, it will go back to the liver. The liver will filter this blood, and it will remove all of these toxins and return the blood as it is back to the heart through the inferior vena cava. Then, regulation of amino acids. The production of proteins is dependent upon amino acids. We all know proteins are made by amino acids, so the liver makes sure that amino acid levels in the bloodstream remain healthy. So it's making the proteins itself, which is albumin, and also the building blocks of proteins, which are the amino acids. You can remember the function like this. So it's making albumin, and it's making the the main ingredients to make albumin, you can say, right? So we have amino acids. Then blood clotting. So you have blood clotting. All the blood coagulants are created within the liver using vitamin K, and these can only be absorbed with the help of bile, which is the fluid the liver produces. So regulation of blood clotting, right? Anything regarding blood clotting is made within the liver with the help of vitamin K. Usually, whenever the liver is damaged, the people have coagulation abnormalities, either increased bleeding and all of that, which you will study, inshallah. Then you have resistance of infection. So we know that the liver is detoxifying, so it's also resisting infections because it's removing other harmful bacteria from the blood, and it's also storing vitamins and minerals. The very famous one we know are the A, D, E, and K, and vitamin B12, as well as iron and copper. So it's storing all of these vitamins and minerals for the body, It has a good amount of store. So even if you are not consuming these vitamins for a short period of time, the liver has enough stores to replenish it and to help your body with the vitamins. Then the processing of glucose. We know the liver removes excess glucose from the bloodstream and it stores it as glycogen. So, and once glycogen is made, the bloodstream will have less amount of glucose. So this is how it's removing the glucose, and it can even convert it back, the glycogen back into glucose. So this is very very important. So it's making glycogen from glucose and it's making glucose from glycogen. So it's just converting them back and forth. So this is the main, functions of the liver, which are important. So you guys can remember the general functions of the liver, and it's very easy to remember. Then next we have jaundice. So what is jaundice basically? Jaundice is the yellowish discoloration, as you can see in the picture, in the eyes of this and the sclera of this patient, and also you can see this old guy over here. He also has, a lot of yellowing on his skin. So, what is jaundice? As I said, yellowish discoloration of the skin, of the sclera, and the mucous membranes due to the deposition of bilirubin. This is primarily due to the deposition of bilirubin. Now, we will discuss how you will have increased bilirubin. There can be different causes. Now, either there can be two things. There can be hyperbilirubinemia. You're making a lot of bilirubin, due to any abnormality, there is increased serum concentration of bilirubin, right? So this bilirubin is depositing and causing jaundice. Or there can be impaired production of bilirubin. For example, cholestasis. Cholestasis is stasis means stoppage, and cholestasis refers to the stoppage of bile. So there can be either impaired production of bile, impaired secretion of bile, or it can be any problem in the outflow of bile. For example, any tumor obstructing the bile duct, or any other causes, preventing the bile to flow out. So, what is the pathophysiology? Pathophysiology is that jaundice is due to an elevated level of serum bilirubin, which may be caused by prehepatic, intrahepatic, or posthepatic defects. Pre means before the liver, intra is within the liver, the problems, and post is after the liver, like for example, within the ducts or the bile duct, the common bile duct. Then the serum bilirubin concentration depends on the rate of formation and hepatobiliary elimination of bilirubin. So any pathology which is causing any abnormality in the process of the bilirubin metabolism, or if it's increasing the bilirubin levels in your serum, it can cause jaundice. Right now, jaundice, as I said, it can affect your sclera. You can cause it scleral icterus, which is the yellowish discoloration of the sclera, and it usually develops at 2 to 3 milligrams per deciliter of more than 2 to 3 milligrams deciliter of bilirubin in your blood, right? So this is once your sclera is being affected. For skin symptoms to appear, or the skin discoloration to become apparent, the serum bilirubin must be at least 4 to 5 milligrams per deciliter of blood. Then other features associated could be pale clay-colored stools, right? Darkening of urine. This is very important. Pruritus, itching, and there can be malabsorption or steatorrhea. Okay, so as I said, it can be prehepatic, intrahepatic, or posthepatic. So prehepatic means, for example, you have a lot of hemolysis. In G6PD deficiency, you have spirocytosis, these diseases. Hemolysis means a lot of breakdown of red blood cells. So red blood cells are being broken down. A lot of bilirubin is being produced, and this bilirubin is in your serum, and the liver is not, it's overwhelming for the liver to conjugate all of this bilirubin, and this bilirubin is just being deposited in all of, in the skin, as I said, in the sclera. So it's causing a problem. Or it can be ineffective erythropoiesis, right? In thalassemias, pernicious anemia, ineffective erythropoiesis means that there is ineffective formation of blood, red blood cells. So once there are these two causes, these come under prehepatic, these will increase the bilirubin, serum bilirubin levels and result in jaundice. Then you have the intrahepatic causes. You have the non-obstructive biliary diseases. Sorry, guys. And then you have the mechanical biliary obstruction. So the non-obstructive, there is either hepatitis, cirrhosis of the liver, primary biliary cirrhosis, or cystic fibrosis. Hepatitis is the inflammation of the liver. Cirrhosis of the liver is formation of yeah. So cirrhosis of the liver is the fibrous scar formation, and okay. So regarding the mechanical biliary obstruction, you have the tumors of the liver. There can be any tumor within the liver, or there can be intrahepatic gallstones, or there can be congestive hepatopathy. All of this. Then the post-hepatic ones are the problems in the either the common bile duct or the cystic duct, right? So there can be any problem. For example, there can be inflammation in primary sclerosing cholangitis. There can be malformation of the biliary tract, congenital problem. Choriodocolithiasis is a stone within the common bile duct, right? All of these. Or there can be a bile duct stricture. All of these are post-hepatic and they are obstructing the bile flow, which is resulting in increased serum bilirubin, and this increased serum bilirubin is resulting in jaundice. So these are just how we differentiate between prehepatic, intrahepatic, and posthepatic. Intrahepatic is anything related or anything, any abnormality of the liver. So we have hepatitis, inflammation of the liver, cirrhosis of the liver, fibrous scar formation, or you have primary biliary cirrhosis is the intrahepatic bile ducts being involved and fibrosis of them, right? Or there can be cystic fibrosis. Then you have the mechanical biliary obstruction, which are the tumors of the liver. There can be any tumor within the liver. There can be intrahepatic gallstones, right? There are stones within the intrahepatic ducts, or there can be primary sclerosing cholangitis. All of these are intrahepatic. Then the posthepatic, I already told you guys. Then we have the gallbladder. Now, regarding the gallbladder, you guys just give me 5 minutes. Okay, so starting with the gallbladder. Sorry guys for the interruption. Actually, I have a throat infection, so it's hard for me to speak for a long period of time. I also have cough with it, so I need to close my mic for that. Okay, so regarding the gallbladder, we have the biliary system. What is the biliary system? The biliary system consists of the hepatic ducts, the gallbladder itself, the cystic duct, and the common bile duct. So the hepatic duct is the right and the left hepatic duct, which I will show you in the picture, and they will join to form the common hepatic duct. So the right duct from the right lobe, the left hepatic duct from the left lobe of the liver, and they are joining to form the common hepatic duct. Common hepatic duct is usually 3 centimeters long, and this is the important thing you need to know about that. Then regarding the gallbladder, we know it has a fundus, a body, and a neck, which I will show you in the next picture, and it lies in the fossa on the inferior surface of the liver. So it's at the inferior portion of the liver, or you can say behind of the liver and inferiorly located. and it acts as a reservoir of bile. So it's the liver is making the bile, and the gallbladder is storing the bile. Then you have the cystic duct. The cystic duct is the duct coming from the gallbladder, and it is also 3 to 4 centimeters long. It usually joins the common hepatic duct to form the common bile duct. Yeah, and that's about it. Then we have the common bile duct, which I will show you in the next picture. This may just skip it. So over here, you guys can see the cystic duct, the right hepatic duct, the left hepatic duct, the common hepatic duct, and then the common bile duct, which is formed by joining all of these together. Yeah, so over here you can see this is the cystic duct, this is the right hepatic duct, the left hepatic duct forming the common hepatic duct. And then the cystic duct and the common hepatic duct will join to form the common bile duct. This common bile duct will come and it will empty its contents into the duodenum at the major duodenal papilla, right? So, and the common bile duct is also joined by the main pancreatic duct. Both of these ducts are emptying their secretions into the duodenum because the food is coming from the stomach and it's highly acidic, right? So the bile also has bicarbonate. So the bile is necessary for neutralizing this food so that it doesn't damages the mucosa of the lower GI tract, right? So this bile is coming and it's neutralizing the acid and it's helping in the emulsification of fats so that we can absorb them. And it is joined by the pancreatic duct with the pancreatic enzymes, pancreatic lipase, pancreatic amylase, all of the pancreatic enzymes which are necessary for the digestion. Then let's move on. This is another picture, just it's more magnified. So you can see the common bile duct, right? You can see the pancreatic duct. This is the pancreatic duct. This is the common bile duct. Both of these are joining, and they are, emptying their contents into the duodenal wall or the major duodenal papilla. So I told you the gallbladder has three parts: the fundus, the body, and the neck. So the fundus is the part which is the downward parts or the expanded parts which is projecting downwards. The main, you would say, the part of the gallbladder. Regarding that, then you have the body of the gallbladder. It is directed upwards, backwards, and to the left. And then you have the neck of the gallbladder, which is just near the cystic duct. So these are the three parts of the gallbladder. You can remember the anterior-posterior relations. So the fundus is anteriorly related to the abdominal wall. Posteriorly, it's related to the transverse colon. The body is anteriorly in contact with the liver, and posteriorly, it's related to the transverse colon. And the neck is the constriction, which is, I told you, near the cystic duct. And its mucosa has oblique ridges, just like a spiral valve. So regarding the blood supply, we have the arterial and the venous blood supply. The arterial, the gallbladder is supplied by the cystic artery. You can see the cystic artery, which is from the right branch of the hepatic artery. So you remember the hepatic artery, right? From the celiac trunk. We said the abdominal layer, the celiac artery, and then the common hepatic artery and the hepatic artery proper. So from there, we have the cystic artery. And the venous supply is the gallbladder is basically drained by the cystic vein. So it's very simple: cystic artery and cystic vein. And the cystic vein is draining into the right branch of the portal vein. Right. So if they ask you a question in the MCQs, where is the cystic vein draining? Into the right branch of the portal vein. So it's very simple. You just remember the main vein, which is the portal vein, and the cystic artery is a right. It's the branch of the right. It's from the right branch of the hepatic artery. So that's how you remember that. Regarding the histology, the lining or the mucosa or the epithelium, the innermost part, it is columnar epithelium, simple columnar epithelium. Then the mucosa or the epithelium is also called the mucosa. It's the same thing. You can call it either the epithelium or the mucosa. Or you can say the mucosa is composed of the epithelium, which is simple columnar epithelium. It basically has mucosal folds. You can see in the picture, these are the folds inside. And it has short and apical microvilli. And these microvilli are good for absorption and increasing the surface area, just to increase the surface area. Then you have the lamina propria. The lamina propria is just below the epithelium, and it is basically, you can also call it the submucosa. So first we have the mucosa, then the submucosa. Sub means under the mucosa. That's why its name is submucosa. It basically has the fenestrated capillaries, the small venules, and it does not has any lymphatic vessels. This is very important. So what does it contain? The fenestrated capillaries and the small venules, but it does not have any lymphatic vessels. And in the neck, this the same lamina propria, it has the mucin secreting glands. The same lamina propria, when you will move towards the portion of the neck, it has the mucin secreting glands, right? Then you have the muscular layer, which is very important of the contraction of the gallbladder and expulsion of the bile from the gallbladder and into the common bile duct, right? So the muscular layer is very important for that. It is for the muscular contraction. Then we have the thick layer of dense connective tissue, which is just after the muscular layer. And at the end, you have the serosa, or the outer covering. So adventitia and serosa, where the gallbladder is attaching to the liver, this is the adventitia. So you can see this is the gallbladder, and this is the part where it is attaching to the liver. This is the adventitia, and the unattached surface is the serosa. This is very important. Adventitia and serosa are two names of the same thing. It's just about their location. So if it's attaching to the liver, the part where it is attaching to the liver, it's called the adventitia. And the unattached part, which is not attaching to the liver, is the serosa. Now, regarding bile, what is bile? Bile, we all know, is a secretion which is made by the liver, and it's stored in the gallbladder, which is helping us in digestion, neutralization of gastric acid, fat absorption, and excretion of bilirubin and cholesterol. So bile is composed of two things, very important: bile pigments and the bile acids. We have two pigments. We have bilirubin, which is the yellow pigment, and then we have biliverdin, which is the green pigment, which is the byproduct of hemoglobin breakdown. Then we have the bile acids. We have the primary bile acids and the secondary bile acids. Now, these bile acids, the primary ones are important. These primary ones are made within the liver. We have the cholic acid and the quinodeoxycholic acid, and they are stored in the gallbladder. And then we have the secondary bile acids. We have the deoxycholic acid and the lithocholic acid. The cholic acid is basically converted into deoxycholic acid, and the quinodeoxycholic acid is converted into lithocholic acid. And these secondary bile acids, they are not made within the liver. They are made within the intestinal lumen by the action of bacterial flora. This is very important because once you have the primary bile acids, right? These bile acids, they are conjugated with glycine or taurine, so they become bile salts. These bile salts will go into your intestines, and these bile salts, they have your primary bile acids. These primary bile acids are then converted into secondary bile acids. Now, very important is that the bile acids are conjugated with the help of glycine or taurine, and these bile acids, they then form bile salts and they become water soluble. So before they are not water soluble. Once they are bile salts in the form of bile salts, then they become water soluble. Okay, so bilirubin, I told you it's excreted as water soluble or conjugated bilirubin. The liver is conjugating the bilirubin, and it's transported in the blood bound to carrier proteins. And as I said, it can be albumin, which is binding this bilirubin. And if it's unconjugated, then it's lipid soluble. Okay, so now I have a table in these slides which will be very helpful in telling you in tracking all of the bilirubin metabolism. So let's just first go through the bile's function. We know that its function is emulsion and absorption of fats, including fat-soluble vitamins, vitamin A, D, E, and K. Then excretion of bilirubin and cholesterol. Uh, this is the majority of the cholesterol is removed by this process, and it has also a very mild antimicrobial effect. Now, regarding the enterohepatic circulation, entero means the intestines and hepatic means the liver. So the circulation is which is going from the intestines towards the liver. Now, the substances that are metabolized in the liver are excreted into the bile. So, as I said, the bile salts and bilirubin. Bile salts, out of them, 95% of the bile acids are reabsorbed back into the liver to be recycled. And they pass into the intestinal lumen where the bacteria will break them down. As I told you, the primary bile acids are broken down how by the bacteria of the intestines. So first they were bile acids, then they combined with glycine or taurine, they became bile salts. These bile salts are then broken down into secondary bile acids, and they can get reabsorbed back again into the intestinal mucosa, and they return to the liver. These bile salts, they return to the liver. As I told you, 95% of the bile acids and the salts will be reabsorbed back into the liver. Then you have bilirubin. Most of this is excreted in the feces as stercobilin, and approximately 20% enters into the enterohepatic circulation, and 90% is recycled in the liver, and 10% is excreted in the kidney. So I will show you a picture. It will be very easy after that. Then we have the bilirubin metabolism. We all know we have the unconjugated bilirubin, which is insoluble in water. It is released into the blood by the macrophages because the macrophages are metabolizing the old red blood cells from the spleen, the splenic macrophages specifically, and then it binds to the albumin and it reaches the liver. Once it's in the liver, it gets conjugated via which enzyme? UDP, glucuronosyl-transferase. This is the enzyme. It conjugates it with what? With glucuronic acid. So the unconjugated bilirubin is joined with glucuronic acid by the help of UDP, glucuronosyl-transferase, and now it is called conjugated bilirubin, or you can call it water-soluble bilirubin or direct bilirubin. Indirect is the unconjugated bilirubin. So the most of the conjugated bilirubin is excreted into the GI tract via bile, right? So as I told you, all of the conjugated bilirubin, where is it present? In the bile. And where is the bile going? Into the GI tract. From the common bile duct we have, it's going into the duodenum. From the duodenum, it will go into the jejunum and all of the small intestines and all of that. Then the conjugated bilirubin is basically which is coming from the bile, which was excreted in the bile. Once it reaches the GI bacteria, it is converted into urobilinogen. This is a compound made from conjugated bilirubin. Now, most of the urobilinogen that you have is converted into stercobilin. Stercobilin is the component or the pigment which is excreted in the feces, and it gives the color to the feces. Now, after stercobilin, some part of the urobilinogen, I told you that the urobilinogen, most of it is converted into stercobilin. Some part of it is oxidized to urobilin. It goes into the kidney, and this gives the urine its yellow color. So these are the two things that you need to remember, very important. From urobilinogen, you get two things, stercobilin and urobilin. Stercobilin is the majority of the urobilinogen is converted into stercobilin, and some part is converted into urobilin. Stercobilin will give the feces its color, and urobilin, by you can remember that it's excreted in the urine, and it will give the urine its color. Okay, so this is the table through which you can track. It is very easy. Just follow it as I'm telling you guys. So the first step is red blood cell breakdown, right? So these are the whole red blood cells. They are phagocytosized by macrophages, mainly in the spleen, and this releases hemoglobin, right? So you are following it like this, and then this. So these are the steps. So red blood cells are broken down, phagocytosized by macrophages in the spleen, and they release hemoglobin. Now, hemoglobin is HB. It's represented by HB, and it has two parts. We have heme and globin, right? So it's split into heme and globin. All of this process is going within the splenic macrophages. Just remember that. Then it has its own separate metabolic pathways. What are those pathways? Okay, let's discuss what will happen with globin and what will happen with heme. Globin is the proteinaceous part, right? So if it's the proteinaceous part, it will be broken down into amino acids, and it will be reused in the body for protein synthesis. Then we have the heme part. The heme part is, we have the porphyrin ring. I hope you guys study the structure, but just for an overview, the heme part is the iron part. So it contains the iron and the porphyrin ring. So the porphyrin ring Iron, what happens with iron? So we told what is happening with globin. Now the next part is the heme. Heme is the iron part. So iron is removed by heme oxygenase. So iron will be removed from heme by the enzyme heme oxygenase, and the ring portion of the molecule. So we have a ring portion to which iron is attached. Iron is removed by heme oxygenase, and the ring will be converted to biliverdin. This is very important. And iron which you are getting is in iron plus 2 form, right? The 2 plus form. It can be either recycled or it can be stored as ferritin, so that if you are not consuming iron, it can be reused again from the ferritin. Then the ring portion, which was converted into biliverdin, this biliverdin is converted into bilirubin. This is where the bilirubin is coming from. Where is the bilirubin coming from? The biliverdin is converted into bilirubin by biliverdin reductase, and this forms the unconjugated bilirubin. Now, once you have the unconjugated bilirubin, you will take this unconjugated bilirubin to the liver. Within the liver, what will happen is that this will bind to the albumin in the blood, right? Within the blood, it's binding to albumin so that it can get transported. So I told you unconjugated bilirubin is insoluble in water, so it cannot be transported in the blood. It needs some transport protein. The transport protein is albumin. So it will join with albumin, will go to the liver, and it will be delivered to the liver. After it's being delivered to the liver will start its conjugation process. It will conjugate the bilirubin. So unconjugated bilirubin will be conjugated with the help of UDP glucuronyl transferase, and it will be conjugated with glucuronic acid. So once this conjugated bilirubin is made, it is water soluble. The water soluble bilirubin is then excreted into the bile or the conjugate. So I told you bile had bilirubin, right? That bilirubin was conjugated. Where did it come from? So I just told you guys, from biliverdin, biliverdin into bilirubin. Bilirubin is then conjugated, taken to the liver, and then the liver conjugates it and excretes it into the bile. And then bile, where is the bile going? The bile is secreted into the intestines. Where in the intestines? As I said, in the second part of the duodenum, in the second part of the duodenum, right, with the pancreatic duct. Now it is preparing for the bacterial action. This bile, okay. Then intestinal processing takes place. The bile is going into the intestines and it's being used up. It has conjugated bilirubin. This conjugated bilirubin is now converted by the intestinal bacteria into urobilinogen. Now this urobilinogen, some of it is excreted in the feces, as I said, as stercobilin, and some of it is reabsorbed and converted into urobilin and goes to the kidneys and goes to the urine and forms the yellow color of the urine. Then we have the final excretion, the same thing as I told you guys, the urobilinogen, stercobilin, forming urobilinogen, and then stercobilin forms the brown color of the feces, and the urobilin forms urine, yellow color, and these end products are eliminated from the body. So this is just a diagrammatic representation. You guys can follow this very easy. This is the heme, the macrophages, unconjugated bilirubin coming to the liver, getting conjugated, getting secreted into the bile. The bile is the conjugated bilirubin is then being getting unconjugated, and by the intestinal bacteria, it's being converted into urobilinogen, and from urobilinogen we have stercobilinogen and then stercobilin, and some of it will be reabsorbed back into the kidneys, form urobilin, and urobilin will be secreted into the urine. This is the main picture which is going on. So this is a picture from first aid, just highlighting the main points. You guys can go through this, very important, and this just tells you the main crux of any process or any topic. So it's very good and very productive. You guys can go through this. It's very helpful. This is again a picture of bilirubin metabolism. Red blood cells, heme, unconjugated or indirect bilirubin is then transported with the help of albumin, and this unconjugated bilirubin and albumin goes to the liver by the help of UDP enzyme. It will be conjugated with glucuronic acid, and then it goes into the bile, and all of that just I told you guys. So this picture is very important. So we have this regulator, source, and effect on bile secretion. So what happens is that how is the bile secretion is regulated, right? So regulation is by the hormone cholecystokinin. So very important is that the parasympathetic nervous system is stimulating the release of bile. So it is the friend of the gastrointestinal system, the parasympathetic nervous system. So if you talk about the cholecystokinin, cholecystokinin is the source is the duodenum. What is it doing? Cholecystokinin, it is contracting the gallbladder. How is it contracting the gallbladder? You guys remember I told you there was the muscular layer within the gallbladder. By that, it is contracting, and it relaxes the sphincter of Oddi, or the sphincter of Oddi, which is the sphincter at the junction at the second part of the duodenum where the common bile duct is emptying its. Sorry, not that junction. The sphincter of Oddi is the sphincter of the gallbladder. Yeah, of the one which empties into the small intestine, right? So then we have secretin, which is the source is the duodenum, and it stimulates the bile production within the liver. Then we have the vagus nerve, the parasympathetic nervous system. From the parasympathetic nervous system, it will also enhance the bile production and gallbladder tone and the bile salts, which are being recycled, as I told you guys, in the bilirubin metabolism. And once they are being reabsorbed and they are being recycled from the ileum and they are going to the liver, they are stimulating further bile secretion and stimulating intrahepatic circulation. So this is again a picture. You guys can follow. It's very easy. The same concept of the bilirubin metabolism and heme metabolism. So that's it. If you guys have any question. So I think. So are you guys listening?